In this section of the teaching presentation, I will be discussing repression. This is a negative feedback mechanism in the tryptophan operon. The repressor protein is produced upstream on the TRP operon by a repressor regulator gene called TRPR. The repressor protein is expressed constitutively at a low level. This means it is produced in relatively constant amounts in all cells of an organism, without regard to cell environmental conditions, so whether there are low or high levels of tryptophan in the cell. I will now discuss how this repressor protein comes into play when there are these low and high levels of tryptophan. Tryptophan can be present in the cell in low amounts. In a low TRP environment, the RNA polymerase binds with the promoter and begins to transcribe the 5 structural genes into mRNA. These can then be translated into enzymes. These enzymes are used to make tryptophan for tryptophan biosynthesis, as previously discussed by Connor. This means in low TRP environments, more TRP is made. E. coli need this tryptophan because it is an amino acid that makes proteins. It can also be noted that the repressor protein is still present but inactive when there are low levels of TRP. It is only used in a high TRP environment. When there are high levels of TRP, the molecule binds to the TRP repressor. When tryptophan is around, it attaches to the repressor molecule and changes its shape, so it becomes active. A small molecule like tryptophan, which switches a repressor into its active state, is called a co-repressor. So, in a high TRP environment, the TRP acts as a co-repressor. This means that it will bind to the repressor and essentially activate it. This will change its shape so it can attach to the operator in the operon. Once it is attached to the operator, the RNA polymerase can no longer bind and carry out transcription. This is because it physically gets in the way of where the RNA polymerase would bind to. This makes sense because if you're in an environment with a lot of tryptophan, there's no need to create more of it. This would be a waste of energy. In this system, the TRP repressor acts as both a sensor and a switch. Firstly, it senses whether tryptophan is present at high levels, and if so, it then switches the operon into the off position. This prevents unnecessary biosynthetic enzymes from being made. Repression isn't essentially the only feedback mechanism in tryptophan. There is also another environmental situation called attenuation, as Amy will now discuss.